Welcome to the SWAYAM NPTEL course on food microbiology for safe and sustainable food systems. In the last classes, we have seen the diseases caused by food-borne microbes, and we have covered the food intoxication and food infections, and now toxico-infections. And we will be covering introduction, difference between infection, intoxication and toxico-infection, and types of food-borne toxico-infection caused by Aeromonas hydrophila, Bacillus cereus, Clostridium perfringens and Vibrio cholera. Microorganisms grow in very huge numbers in food. Gastroenteritis occurs due to ingestion of a large number of viable cells of pathogenic bacteria through food and water. Generally, toxico-infections involve either sporulation or lysis of bacterial cells and release toxins from lysed cells in gastrointestinal tract. The following are some of the characteristics of food-borne toxico-infections. For gram-negative non-spore forming bacteria, ingestion of viable cells in large numbers is necessary together with food. The cells rapidly multiply in the intestinal tract. Many cells lyse and lysed cells release toxins. For spore forming bacteria, ingestion of large numbers of live vegetative cells is necessary together with food. Vegetative cells of spore formers do not multiply in digestive tract, but they can sporulate and their lysed cells release toxins. Toxins of both groups can cause gastroenteritis symptoms. Food-borne toxico-infections are caused by Aeromonas hydrophila, Bacillus cereus, Clostridium perfringens, enterotoxigenic Escherichia coli, Plesiomonas shigelloides, Plesiomonas shigelloides and Vibrio cholera. Some of the important food-borne toxico-infections are included in this class in alphabetical order based on relative importance of the disease, characteristics of microorganisms, predominant types of food, mechanism of disease, prevention and control of pathogen, and analysis of illness. Now, the difference between infection, intoxication and toxico-infection. Infection occurs when a harmful microorganism is ingested and grows in gastrointestinal tract. Examples include food-borne viruses and parasites and bacterial infections like Salmonella. Symptoms include diarrhea, nausea, vomiting, abdominal cramps, headache, fever and chills. Intoxication occurs when a harmful toxin produced by the microorganism is ingested. Examples include bacteria like Clostridium botulinum and Staphylococcus aureus. Symptoms include nausea, vomiting, abdominal cramps and diarrhea. Toxico-infection occurs when a harmful microorganism is ingested and produces a toxin in the human body. Examples include bacteria like Clostridium perfringens, Bacillus aureus, enterotoxigenic Escherichia coli, and Vibrio cholera. Now, types of foodborne toxico-infection. First is Aeromonas hydrophila. Characteristics of bacterium. Aeromonas hydrophila is a common inhabitant in seawater where the salt concentration is about 6.5% or more, fresh water and estuarine water environment during the summer. It is pathogenic to fish. It also occurs in the intestinal contents of humans and animals. It belongs to Vibrionaceae family and is a gram-negative bacilli. It can produce capsule if it grows in glucose-rich medium. The high levels of Aeromonas hydrophila can also associate with food spoilage and may produce pungent off odors. It can produce mildly heat resistant extracellular lipase and protease enzymes. Nature and association with food. Aeromonas hydrophila is ubiquitous in water environment. It is not normal habitat of gastrointestinal tract of human. It is common in seafood including fish and shellfish. In meats, poultry and milk, this bacterium is generally present in low numbers, but may greatly increase during storage at chill temperatures. A wide variety of vegetables including spinach, celery, bean sprouts, broccoli, lettuce and root vegetables may contain Aeromonas hydrophila from contaminated water. It does not survive at mild cooking temperature, but may be introduced at post-process contamination from uncooked and contaminated water. Therefore, unheated and recontaminated ready to eat foods may pose a greatest health risk due to Aeromonas hydrophila illness. Disease and symptoms. Infection with Aeromonas hydrophila can cause wound and systematic illnesses in humans. Infection from open wounds may lead to tissue infections and septicemia. Foodborne illness, that is gastroenteritis, results from ingestion of a sufficient number of bacterium in foods and water. Gastroenteritis most commonly occurs in infants and children under 5 years old, elderly and immunocompromised people. Symptoms appear after 24 hours following ingestion of foods containing viable Aeromonas hydrophila. The infective dose of Aeromonas hydrophila ranges from 10 to the power 4 to 10 to the power 10 viable cells per gram of food or milliliter of water. Aeromonas hydrophila may survive in stomach and pass to intestine. The main virulence factor, that is the aid pathogenicity of Aeromonas hydrophila, associate with the production of adhesion that is the fimbriae, exotoxins, enterotoxin, beta hemolysin or aerolysin, heat stable and heat liable Shiga-like toxin, extracellular enzymes such as protease, lipase, phospholipase, elastase and DNase, and endotoxins, that is lipopolysaccharide. Aeromonas hydrophylla attaches to the intestinal epithelial cells by fimbriae. Aeromonas hydrophylla produces enterotoxin and lyse cells release endotoxins. Toxins cause fluid secretion from epithelial cells. The most common symptom is watery diarrhea. Other symptoms include fever and vomiting. Generally, disease is self-limiting and lasts within 2 weeks. A second type of gastroenteritis of Aeromonas hydrophylla is a dysentery-like illness characterized by diarrhea containing blood or mucus and abdominal pain. Prevention and control. Additional prevention method can be used for foods stored at low temperatures, that is below 5 degrees Celsius. Sufficient cooking of seafood and disinfections of drinking water prevent Aeromonas hydrophylla illness. Polyphosphates and sodium nitrite can also control the growth of bacterium in foods. It grows under modified atmospheres. It can be controlled by heat treatment, thus preventing post-heat contamination and using one or more growth limiting parameters such as low pH and low water activity. Vegetative cells of Aeromonas hydrophylla are easily destroyed by heating at 56 degrees Celsius for 10 minutes. Aeromonas species produce heat stable exotoxins and exoenzymes that may survive this heat treatment. Now, the Bacillus cereus. Characteristics of bacterium. Bacillus cereus is gram positive rod shaped, that is 1 to 2 micrometer width and 4 micrometer in length, motile and aerobic bacterium. It produces central to terminal ellipsoidal or cylindrical endospore and does not cause swelling in the cell. Motile Bacillus cereus has peritrichous flagella, but non-motile strains can also present. It grows at a temperature of 10 to 50 degrees Celsius, optimally between 28 to 35 degrees Celsius and pH of 4.9 to 9.3 and tolerates NaCl up to 7.5%. It is unable to grow at temperature below 4 degrees Celsius. However, cyclotrophic strains are present and can grow at temperatures as low as 6 degrees Celsius. Nature and association with food. Spores and cells of Bacillus cereus are widespread in nature, common in soil and dust and in vegetables, meats, eggs, cereal grains, rice, flour, pasta and dairy products are the most common food vehicles for Bacillus cereus. When pasteurized milk and cream are stored at inadequate chill temperatures, Bacillus cereus grow and cause spoilage known as sweet curdling or bitty cream. Dried herbs, spices, vanilla, cereals and flour can be important sources of Bacillus cereus, and they increase incidence of Bacillus cereus food poisoning. Diarrhea illness mostly associates with eating heavily contaminated food, including meat products, milk based puddings, dried milk and sauces. Bacillus cereus easily spreads to foods, especially those of plant origin, for example rice and pasta. Now, disease and symptoms. Its presence in foods is not a significant health hazard unless the bacterium is able to grow and produce toxins in foods. Bacillus cereus causes two types of food poisoning, the emetic and diarrhea illness. The characteristics of two types of Bacillus cereus foodborne diseases are described in table. Foodborne for both types, food involved has usually been heat treated that allow spore germination to vegetative cells. Bacillus cereus grows well after cooking and during cooling of foods. Heat treatment can also activate spores, and Bacillus cereus grows well in the absence of competing flora. Bacillus cereus foodborne illnesses are relatively mild and usually last less than 24 hours. This table shows the characteristics of two diseases caused by the Bacillus cereus, that is diarrheal illness and the emetic illness. The infective dose for diarrheal illness is required 10 to the power 5 to 10 to the power cells per gram, whereas for emetic illness 10 to the power 5 to 10 to the power 8 cells per gram. The toxins produced by diarrheal illness in small, the toxin produced in the small intestine for the diarrheal type of illness, whereas for emetic illness it is performed in the foods. Type of toxin produced is the protein as enterotoxins in the diarrheal illness, whereas it is cyclic peptides in the emetic illness. The incubation period for diarrheal illness is 8 to 16 hours, whereas it is 30 minutes to 5 hours for the emetic type of illness. Symptoms for the diarrheal type illness is abdominal pain, watery diarrhea and nausea, whereas for the emetic type of illness it is nausea, vomiting and diarrhea. Frequently associated foods for the diarrheal type illness is cooked meat and poultry, vegetables, sprouts, soups, puddings and milk products, whereas for emetic type of illness the frequently associated foods are fast foods, fried and cooked rice, pizza, pastry, noodles, etc. In general, a large number of viable cells, that is up to 10 to the power 5 to 10 to the power cells, that is up to 10 to the power 5 to 10 to the power 7 cells per gram of food, need to be ingested together with food to cause gastroenteritis. Ingested cells survive in stomach and pass to intestine, multiply and produce enterotoxin in small intestine. Vegetative cells sporulate and lice cells release enterotoxin. The diarrhea symptoms appear within 8 to 16 hours. The symptoms include abdominal pain, watery diarrhea and occasionally bloody, nausea and rarely vomiting. 3 different enterotoxins, that is the virulence factor responsible for diarrhea, are hemolysin, which lyses erythrice, cytotoxin and non-hemolytic toxin. These toxins are generally heat and protease sensitive and are produced in the small intestine. Prevention and control. The important factors for Bacillus cereus foodborne diseases are improper handling, improper holding temperature, contaminated equipment, inadequate cooking and poor personal hygiene. Food should be kept at low temperatures to prevent spore germination. Stored foods must be reheated above 75 degrees Celsius before serving. When foods are exposed to sublethal temperatures, spores of Bacillus cereus can be stimulated to germinate. Rapid cooling within 2 hours and proper reheating of cooked food are essential if the food is not consumed immediately. Temperatures under 100 degrees Celsius will allow survival of some Bacillus spores, but steaming under pressure, thorough roasting, frying and grilling are most likely to destroy cells and spores. Now, Clostridium perfringens. Characteristics of bacteria. Clostridium perfringens is the most widespread of all pathogenic bacteria and the most common type of bacterial foodborne disease. There are several toxigenic types of Clostridium perfringens like A, B, C, D and E based on the production of enterotoxins, that is heat labile, like alpha, beta, epsilon and iota. Clostridium perfringens type A produces only alpha toxin. Type B produces alpha, beta and epsilon toxins. Type C produces alpha and beta. Type D produces alpha and epsilon and type E produces alpha and iota. Toxins are produced during the exponential growth and sporulation of bacterium. Clostridium perfringens type A is responsible for food poisoning and gas gangrene in human. Clostridium perfringens is a gram-positive, large, rod-shaped, up to 2 to 4 micrometer in length, anaerobic, endospore forming, that is oval, central or sometimes subterminal, swelling with spore, capsulated, non-motile, mesophilic and catalase negative bacteria. Growth occurs at 14 to 51 degrees Celsius with optimum at 43 to 55 degrees Celsius with optimum at 43 to 45 degrees Celsius. Vegetative cells grow with pH ranging from 5.5 to 8.3 with optimum being 6 to 7. The minimum water activity ranges from 0.97 to 0.93. Vegetative cells of Clostridium perfringens are sensitive to low heat treatment, that is the pasteurization temperature, but their spores are extremely heat resistant and some can survive even at boiling temperature for several hours. Spores of food poisoning isolates generally have considerably greater heat resistance than spores of Clostridium perfringens isolates from other sources. Nature and association with food. It is a common inhabitant of the intestinal tract of healthy animals and humans. The presence of bacteria in soil and feces provides opportunity to contaminate foods. Due to its ability to grow over a wide temperature range, it is often implicated in human food poisoning. Large volumes of food provide an anaerobic environment and temperature for germination of spores and outgrowth. The most common food vehicles for Clostridium perfringens illness are meats such as beef and poultry, and meat containing products such as gravies and stews. The bacterium may be carried into the kitchen through meat dishes or hands of food handlers, as well as through many other foodstuffs. Disease and symptoms. Clostridium perfringens can cause two types of gastroenteritis and gas gangrene. Clostridium perfringens type C can cause necrotic enteritis, that is pig bell with beta toxin, and Clostridium perfringens type A causes the second gastroenteritis with enterotoxin. The infective dose required for initiating this disease ranges from 106 to 107 viable vegetative cells per gram of food. Clostridium perfringens multiply and sporulate in the gastrointestinal tract. Enterotoxin is produced during the sporulation of Clostridium perfringens cells in the small intestine, and enterotoxin is released upon lysis of cells. Enterotoxin binds to plasma membrane of the intestinal epithelial cells and causes morphological changes. The symptoms appear within 8 to 24 hours after ingestion of food containing large numbers of viable cells. The most common symptoms are watery diarrhea, nausea, mild abdominal pain, chills, headache, less commonly vomiting, and rarely fever. Factors contributing to foodborne diseases by Clostridium perfringens are inadequate cooking temperatures allowing the survival of spores of Clostridium perfringens, pre-cooking of food the day before serving, inadequate and prolonged cooling of cooked meat containing small numbers of surviving spores. Germination of spores and multiplying rapidly to produce a large vegetative population at room temperature during the time between cooking and surviving. Refrigeration of large portions of previously cooked foods. Inadequate reheating, and finally the storage of foods such as meat dishes at warm temperature, that is below 60 degrees Celsius, for long time after cooking. Prevention and control. The Clostridium perfringens foodborne disease can be prevented by reheating cooked chilled foods to a minimum internal temperature of 75 degrees Celsius immediately. Reheating cooked chilled foods to a minimal internal temperature of 75 degrees Celsius immediately before serving, so that vegetative cells are destroyed. Keeping meat above 60 degrees Celsius after cooking. Cooling cooked foods rapidly. Serving cold food held below 10 degrees Celsius for 2 hours. Educating food handlers about hygiene practice. Controlling this bacterium in food services with strict time temperature control. Effective sanitary care of equipment, utensils and workers' hand. Using proper sanitation during preparation and handling of foods, and finally cooking of foods to the highest temperature, that is above 75 degrees Celsius, to kill cells as many spores as possible. Now, the Vibrio cholera. Characteristics of bacterium. Vibrio cholera serotype 01 and non-serotypes 01, 0139, Ogawa, Inaba and biotype E1 Tor cause cholera. Vibrio cholera is a member of family Vibrionaceae. It is gram negative, motile and polar flagella. Motile with polar flagella, curved rod, catalysin, oxidase positive and facultative anaerobic bacterium. The optimum growth temperature is 35 degrees Celsius, with the growth temperature ranging from 15 to 42 degrees Celsius. It can multiply in contaminated seafood and rapidly grows in cooked seafood. It survives in foods at 5 to 10 degrees Celsius and growth pH range from 6 to 10. Alkaline foods facilitate rapid growth. Nature and association with food. Vibrio cholera is widely distributed in aquatic environments. Cholera is primarily a waterborne illness. The main source of Vibrio cholera is the infected people. Cholera results from the ingestion of infective Vibrio cholera through food and water contaminated with feces of people suffering from illness. Direct fecal contamination of cooked foods can also serve as a route of transmission, particularly if foods are left at ambient temperature for a period of hours after contamination. The most common food associated with cholera are crabs, shrimps, raw fish, oysters, cooked rice, frozen coconut milk, raw vegetables and fruits. Now, disease and symptoms. Vibrio cholera colonizes on the small intestine epithelial cells by means of one or more adherence factors such as pilus. It can multiply rapidly to produce cholera enterotoxins, which is a protein by nature, on epithelial cells. When the bacterial cells are lysed, the enterotoxin is released into the intestinal lumen. Cholera toxin disrupts ion transport of epithelial cells and stimulates adenyl cyclase. Initially, the diarrhea is brown and then becomes pale grey with slightly fish odor. Mucus in diarrhea imparts the characteristics rice water appearance, that is rice water diarrhea. Symptoms follow with prostration. Symptoms follow with prostration, painful muscle cramps, anorexia, abdominal pain, clouded mental status, loss of fluids in many liters per day, and dehydration of intestinal cells in short time. Vomiting may also be present. Bloody diarrhea occasionally occurs. Cholera pathogens. Cholera pathogenesis, that is virulence, involves cholera enterotoxin or pilus, which mediate colonization in the small intestine. Cholera toxin is encoded by chromosomal genes. Preservation and control. Vibrio cholera strains are sensitive to heat and killed by temperatures used for cooking. The spread of disease can be prevented or reduced by proper hygienic practices. Contamination of food and water directly or indirectly by fecal matter should be prevented. This can be achieved by developing effective sanitation in water supplies and treatment of water and disposal of sewage. Low temperatures, high organic compound content, high moisture and absence of competing flora enhance survival and growth of Vibrio cholera in food. Cooking eliminates many competing microorganisms, may destroy some inhibitors, and may produce denatured proteins that allow rapid growth of Vibrio cholera. For example, cooked rice is a good growth medium for this bacterium. Normal food manufacturing processes such as pasteurization, irradiation, low water activity and adequate cooking are enough to eliminate Vibrio cholera.